after 2 hours after a full meal. What is the correct? Which is correct about the structure of?
Okay.
So our answer as a group is
Mature insulin. Okay, go ahead.
yes. Our answer for number 1 is letter D.
Mhm.
1 S to S bond connects A7 to B7.
Okay, so why are the rest A, B, C not correct? You have to discuss that also.
Okay, so
Uh-uh.
ayan, so for letter A, it says
Uh-uh.
combines C-peptide A and B chains.
Why is that not correct? Ala, kayo ha, nag-shortcut kayo ha. Kailangan alam nyo. Eh mamaya yan ang gagawin ko
How?
correct answer. O, di ba? So what makes option A wrong? Anybody? Anybody can jump in. Sinong gustong tumulong kay from the group 1, kay Kimberly? Sabi ko sa inyo, discuss bakit mali yung iba eh. Dumiretso kayo. O sige kayo. Bakit mali ang option A, option B, option C? Ganun
Doc, yun
tayo.
dun sa option A po.
Ah,
Dapat instead of composed of C-peptide, dapat composed po siya of alpha and beta chains. Tapos
okay.
yung C-peptide po yung natrium. And then
Okay.
for choice B naman, yung 30 amino acids should be comprised of the beta chain. And
Ah,
then for option C, yung 21 amino acids po should be comprised of the alpha chain.
okay, very good, no? So, ang ibig sabihin ng C-peptide is connecting peptide. Tama? It is supposed to connect your alpha and beta. But in the in the release of mature insulin, nakahiwalay na siya, chinugi na siya, together with magnesium, no? Therefore, dahil ang half-life ng insulin is very short, what is measured in the laboratory is actually C-peptide. So, 1 C-peptide equals 1 mature insulin. Did you get that? Tumango kayo. Yes or no? Yan. Okay. So, yung B and C, pinagbaliktad ko rin. So, ano yung, pag sinabi kong intra, ilan ang intra-SS bonds? Ilan? If instead of asking for inter, because inter means it is within the polypeptide chain, right? Pag sinabi kong intra, it is connecting alpha and beta. O ilan? Para lahat ng SS chains. Anybody? Don't be shy.
One
Judith,
intrachain po, Doc.
ilan?
One intrachain po, Doc.
1? Agree kayo? Intra, ha? There's supposed to be 2. So, all in all, you have 3 SS linkages. And when you talk about SS linkages, what amino acid contributes to the SS? Meaning to say there is sulfide. Ano? Which one? Pag sinabi kong SS, dapat
Cysteine po,
anong
Doc.
amino acid meron?
Cysteine po, Doc.
Cysteine. Oo, ha? Kasi hindi naman nagko-contribute ang methionine sa SS, no? Iba yung role ng methionine. So, whenever you have an SS bond, ang ibig sabihin nun, ang participant is cysteine. Okay. So, okay, yun lang ang importante. Any questions about the structure of mature insulin? So, dapat alam nyo na insulin is what type of hormone? Group 1, group 2. E1. Ano? Ano klaseng hormone ang insulin? Just like glucagon, starting with letter P. May hint pa, ha?
Pancreatic po.
From polypeptide hormones are synthesized as larger hormones. So, merong pre-pro-insulin, pro-insulin, and then finally, mature insulin. So, you should be able to know what are the differences, the important features. Okay. So, please, what do you call that? Look at the learning outcomes that I posted yesterday. So, okay na tayo. Any questions? Okay, let's go to number 2. Oh, group 2. Paki-explain niya number 2. We're now going to talk about the insulin receptor. Oh, sino ang group 2? Ala, walang may gusto. Baka bukas na tayo abutin dito. Group 2, anybody? Sige na, wag na kayo mahihiya. Oh, Dio John. Go ahead, Dio John. Elarco. Go ahead, anak. Nakamute ka naman. Ayan. Perfect.
Okay.
Oh, sige. Insulin receptor. Oh,
Okay. Which statement is correct about the insulin receptor?
okay.
The correct answer is letter A po.
Okay.
This is because insulin binds to the receptor and the alpha subunits
Okay.
will drive a change in conformation of the beta subunits.
Okay.
So this conformation, there will be autophosphorylation of the tyrosine residues in the
Okay.
beta subunit.
Very good. No? So if you look at the structure of it's okay, darling. It's okay. So, ang ibig sabihin niyan, if you look at the structure of insulin receptor, it is a heterodimer. Ibig sabihin, it is composed of 4 polypeptide bonds. 2 alpha, which are extracellular, diba? Outside the cell, and then 2 beta that are intracellular. They are also joined by your SS bridges. So, meron pa ring SS. So, ang ibig sabihin nun, meron pa ring cysteine residues. Now, what type of receptor is IR or insulin receptor? Nandyan na yung sagot yung autophosphorylate tyrosine residues. What do you call that that big family of receptors? Ha? Parang wala naman akong kausap. Hi. Anybody? Group 2 pa rin. Anong tawag dito sa ano na to? Tyrosine receptor family, no? So if you look at the insulin receptor, it is also an enzyme because it can phosphorylate, which is the same family as your growth factor. Okay? And your GLP-1. Okay. Eh, bakit mali yung A? Anong meron sa A subunit? The one that is above. Outside the cell. Makes sense. Ano dapat ang function niya? Group 2. Anong function ng alpha subunit in contrast to beta subunit? Magtatawag na ba ako? Any volunteers? Wala? Uwi na tayo. Bahala na kayo. Oh, ano? Sino gusto mag-recite? Huwag na kayo mahihiya. So, ganito lang.
Yung sa alpha po, kasi ang kinocontain
Okay,
yung-
ganit. Oo. So, anong meron sa alpha subunit, anak? Bakit hindi
yung
binding?
insulin.
Okay, so you call that your ligand binding site. Ligand is insulin. So any small molecule is usually called a ligand. Now, if you look at the structure of insulin receptor, there are 2 alpha, no? So pwede ba siyang mag-bind ng dalawang insulin? True or false? Pwede, hindi pwede. Pwede ba isa lang? No, sa tingin niyo? Hindi kayo makatingin. Is it possible to bind the two? What do you think? I don't think. Okay, so it's possible to bind two insulin molecules, but one of the alpha subunits is predominantly active over the other. So kahit isa lang yung mababind na insulin, pwede na ma-activate yung signaling pathway ng insulin. And of course, your beta subunit, your C-termini of the alpha subunit binds, it's not that C-terminal, ha? It's the alpha terminal. Kaya mali yan. Did you get it? Did you get it, guys? Okay. So therefore, ang nag-autophosphorylate or the one with active enzyme kinase activity is the beta. But the story is, kailangan i-autophosphorylate niya yung nasa kabilang banger, hindi yung diretso dito. Okay? It will autophosphorylate tyrosine units, critical tyrosine units, on the opposite beta. So, ganon. May cross-phosphorylation. So, yun ang kwenta ng IR. Any questions about IR? So, let me repeat. It is a heterodimer. It's made up of 2 alpha, which are extracellular and are supposed to bind your ligand or your insulin, no? And your beta subunits, which are intracellular inside the cell, and they are supposed to autophosphorylate the opposite critical tyrosine units in order to start the signaling pathway. Maliwanag, ha? Okay, let's go to number 3. Group 3. Paki-discuss on number 3. Ang shy nyo naman. Dali, group 3. Sinong gusto mag-discuss? Anybody? Sige na. Ano ba sinagot nyo? Sumagot ba kayo o wala? Nagchismisan na kayo? Kahit mali yung sagot nyo, okay lang. Group 3. Please lang, oh. Gusto ko na matulog. At sama ng pakilasa ko. Anybody?
Doc, for number 3 po, insulin suppresses
Sige.
the increase of free fatty acids in blood by inhibiting the process. Answer po is letter B, phosphorylation of HSL. Sino ba yung ratio? So for insulin po kasi, yung work ni insulin, since our case po, nasa fed state po yung patient natin.
Okay.
Phosphorylation po of HSL would inactivate yung, hindi po mapopromote ang lipolysis. Therefore, hindi po magkakaroon ng increase in free fatty acids po.
Okay. So, para lang matandaan nyo, dapat walang lipolysis. Well-fed state eh. Tama? Ganun lang kayo mag-isip. Why do you need to break down? You have a lot of free fatty acids coming from the diet and from glucose. O, i-evaluate nyo daw ako. Yan. So, this is one concept that you have to remember, diba? You don't need to stimulate or phosphorylate hormone-sensitive lipase. That is HSL. And when you hear HLSL, para madali nyo matandaan, this is palaging associated with glucagon. Did you get that? Para wala nang kaduda-duda. Pag well-fed state yan, hindi pwedeng glucagon. Tama? So, hindi rin pwede na HSL. Okay. Bakit mali yung ibang choices? Ano yung CAT-PT1? Carnitine palmitoyl transferase 1. Saang metabolic pathway nyo yun na meet sa lipid metabolism?
For beta oxidation po, Doc.
Beta oxidation. So, in the same manner, wala din beta oxidation. Correct? Ayan, ganun kayo mag-isip. Letter C. Ano yung HMG-CoA synthase? Key enzyme siya ng what metabolic pathway? Anybody? Sagot na yan. Sagot.
Ketogenesis po.
Michelle. Michelle,
Ketogenesis po, Doc.
ketogenesis. Wala din, diba? So, ketogenesis usually starts 24 hours in the fasted na, and then gradually it becomes maximum during the starved state. Because at this time, your brain will already start to use ketone bodies. So, that is like 1 week without food intake. Nakuha niyo yun? Tango naman kayo. Sumagot naman kayo.
Po, Doc.
Okay, very good. Thank you. So, ano naman si Pepsi K? Bakit mali siya?
Pepsi K po kasi is for gluconeogenesis.
Okay, very good. Okay. So, So, mammalian target of rapamycin. Rapamycin is an antibiotic na complex 2. So, mTORC1 is usually like a sensing, no? Na nafe-feel niya kung madaming nutrients yung katawan, and it will activate certain metabolic processes that will supply the body with nutrients. No? On the other hand, mTORC2 is more for regulation. So, hindi ko na discuss pa dito kasi it's very complicated. You'll meet them again when you go to second year and third year. Sige. Ano sagot sa number 4? Group 4. Group 4. Are you still there? Sige na. Parang wala na tayong kausap pa. Group 4?
Hi, Doc.
Yeah, go ahead, Anak.
Group 4 po, our answer was C, activation of SREBP1.
Okay. Why? What
this
is
is
SREBP1?
it's a transcription factor that
Okay.
actually upregulates the genes encoding
Okay.
enzymes for fatty acid and triglyceride synthesis po.
Okay, very good, no? Just to give you a guideline and something to remember I remember this one. SREBP1 stands for sterol. So, lipid sterol, diba? Response element binding protein 1. So, it is really a transcription factor. And when you talk about transcription, you're talking about factors that can inhibit or stop the transcription of certain enzymes. So, yun lang yung ibig sabihin nun. Okay?
Okay,
So,
Doc.
yes, continue, Laila.
With what po?
Why is A wrong?
Oh, the activation of PDE3B
Uh-uh.
is activated through phosphorylation by protein kinases like
Okay.
PDA and KB po.
Okay.
For B, it's wrong because the activation of FOXO is activated by the dephosphorylation,
Very good.
which occurs through the inhibition of PI3K or AKT pathway.
Dami, no? Acronyms. Okay. And the
Yes,
last one, LXR.
the inhibition of LXR it's inhibited by various mechanisms, including the binding of special ligands, such as
Okay.
cholesterol.
Sige. Para hindi nyo lang makalimutan, PDE3B is phosphodiesterase. No? So, it is involved with CCAMP, no? Cyclic AMP. FOXO, ano nga ba? Fork head of, ewan ko kung ano pa yan, no? And it is usually involved with lipolysis, which should not occur in the well-fed state. And LXR is more related to vitamin A receptors. Did you get that? No.
Yes, Doc. Thank you.
Yeah, any questions about that? Okay. If I find the shortcut of all these acronyms, I'll send it to you para hindi lang puro ano ang gusto nyo ba? O wag na lang.
Okay lang,
Okay
Doc.
na ba? Okay, sige. Next.
Here.
Yeah, I'll send it to you. Huh? Group 5. Group 5, number 5. Anybody? Shy pa kayo. Number 5. Sino ang sasagot? Sige na. Oh, anybody. Kahit na hindi group 5, sinong gusto magsagot. So, direct activation by phosphorylation of ACT2 and protein kinase C. Ano nga ba yun? Ganon. At saka atypical, no? Is catalyzed by which enzyme? Oh. Sinong sasagot? So, ACT2 is actually a major node, no? In the regulation by insulin. And the other name of ACT2 is what? Anong ibang pangalan ng ACT2? Wala, parang wala na akong kausap pa. What's the other name of
Protein.
ACT2? Yes? Jabi?
Ano po, protein kinase B po.
Ha? I did not hear you. Ha? La, hindi mabasa yung notes nung kapitbahay. Ano?
So, protein kinase B po.
Okay, protein kinase B or PKB, no? So, ang ibig sabihin na meron PKA, PKB, PKC down the line, no? So, ang ibig sabihin yan, PKB, the other name of ACT2 is a kinase. So, what does a kinase do? It's supposed to add phosphate groups. So, nakuha nyo yan? Para lang meron kayong panghahawakan na hindi kayo memorya ng memorya. Okay. So meron din siyang PKA, ano nga ba yun? Gamma and atypical, no? Mga kulot-kulot. So, ang sagot nyo, what's your answer? May sagot ba kayo? Wala pa? Ano sinagot nyo?
Letter B po.
Letter B. Okay. PDK1. Okay. It's one of the major nodes. Okay. Agree kayo, everybody? Bakit hindi A? Map K. Anong trabaho ng Map K? Pag meron kayong mga magiging anak sa future, yan, pwede yan. Meron pang son of 7 less, no? So, Map K, mitogen. Tingnan nyo, kasi mitogen. So, it's associated with, anong mitogenesis? Pag sinabi kong mitogenesis, it's supposed to be associated with mitosis. Okay? So, it is cell proliferation. So, let me just remind you that insulin is not only involved in metabolism, it has other functions, and we call them pleomorphic. And basically, one of the functions of insulin is with cell growth because of MAPK and differentiation. No? So, magkakaiba yung mga biyahe depending on what area of the signaling pathway of insulin is involved, then you'll have different effects. That is the reason why ang dami-daming kaaway ni insulin, no? Dami, no? The counter. But if you look at the signaling mechanism of insulin, it's very complicated. Kasi mag-isa lang siya. Ang kanyang kakampi lang si Incretins. Any question? O, ano naman yung PI3K? Anybody who can tell me what is PI3K? Para maalala nyo na. Ano function ng PI3K? Which is still a kinase, diba?
Ano po, Doc? Catalyzes po yung phosphorylation po ng PIP2.
Okay, so you have, when you look at the cell, there are lipids in the membrane, okay? And the job of PI3K is to phosphorylate PIP2, ang cute ng pangalan na, to PIP3. Na? Walang PIP4, hanggang po PIP3 lang. So that will now also send or activate the other mechanisms. By the way, I forgot, no? The basic first proteins that are phosphorylated upon autophosphorylation of your beta subunits are called your insulin receptors, IR, which you have 4 types, no? 1, 2, 3, 4. But the major insulin receptors that are utilized by insulin is insulin receptor 1 and 2. Did you get that? No? So, hindi pa nila na-discover bakit may 3 or 4. Siguro kayo na mag-discover nun. Okay. Oh. Ano naman si SIC2? Salt-induced kinase 2. Anong function nun? Para maalala nyo na. Saan pathway pag na-activate ang SIC2? Sige na, look at your notes.
Gluconeogenesis po.
Gluconeogenesis. Yan. Ganun lang kasimple. Pag nakita nyo yung pathway, dapat alam nyo kung ano yung stimulated or inhibited. So you don't have gluconeogenesis in the well-fed state. So general statement, insulin is a major anabolic hormone, diba? So it is secreted during the well-fed state, and it is supposed to, what do you call this? Stimulate genesis. Diba? Genesis stands for synthesis, except glycolysis. Because glycolysis is not genesis. But the primary function for eating is to provide energy. So yun yung oxidation of hexoses. Yun ang unang nangyayari. And any excess can now be stored as glycogen, fatty acids, and proteins. O, di ba ang dali? Papatayin nyo na ako eh. Any questions? But number 5. Any questions? So far, so good? Sagutin nyo naman ako. Tango lang kayo.
Po, Doc.
So far, so good. Okay, number 6, group 6.
So for the number 6 po, which
Ah, ah.
enzyme is phosphorylated and inactivated by AKT2, resulting in the storage of excess glucose in liver and muscle after a high carbohydrate meal. So the
Okay.
choices are A is GSK3, B is PI3K, C is PKB, and D is SIC2. Our answer po is letter A.
Letter A. So GSK3 is glycogen synthase kinase 3. O, ano naman ang work ng GSK3? Bakit mo ma-stimulate ang glycogenesis?
So po, after a high carbohydrate meal po, glycogen synthase is stimulated by insulin. This
Okay.
activates AKT2, which
Ah.
phosphorylates and inactivates the enzyme GSK3, thereby enhancing po the glycogen synthesis.
Okay. So, ang direct effect niya actually, when you activate GSK3 by phosphorylation, it cannot inactivate glycogen synthase. Nakuha niya? So, pag active ang glycogen synthase, then glycogenesis will occur. Sabi ni Rene. Anong pinagsasabi mo, doktora? Natatawa na lang kayo. Okay, we talk about PI3K, diba? We also talk about PKB or ACT2 and SIC2. Any questions so far? Christian, may question ka ba? Alam mo na, no? Christian? Ano? Merong learning outcomes akong pinost yesterday. Check nyo lang para kasi that will be the basis. Nakangiti na si Rene. Okay, number 7, group 7.
Doc,
Yes.
rinig po ba ako?
Yeah, I hear you, anak.
Okay po. The activation, so for question 7, the activation of PI3K or AK2 by insulin inhibits which metabolic effect? And ang sagot po dito ay ang inactivation or of bad preventing apoptosis. This is because na-phosphorylate po yung bad BACL XL complex at the serine residues S112 and S126. So kapag na-phosphorylate po ito, nagbabind po siya sa 14-3-3 protein.
Okay.
Kaya nasa-sequester po siya sa cytoplasm, kaya hindi po
Okay.
mag-i-initiate ang apoptosis.
Very good, no? So bad, as the name implies, is accelerating apoptosis. Ano ba ang apoptosis? It's cell. Reni, tawa ka ng tawa na distract ako sa'yo. Hindi ako titingin sa'yo. Apoptosis, apoptosis means its cell has a lifespan. No? So, halimbawa, na-reach, umalis tuloy siya. Na-reach na ng cell yung kanyang taning ng buhay, eh talaga mamamatay na siya. And the factor that is promoting, no? Apoptosis is bad. But it has to be phosphorylated in order to be active. Ngayon, pag dephosphorylated siya, hindi niya go signal yung apoptosis. So basically, this is, what do you call this? This is involved with the beta cells of the pancreas and the other cells. Kaya nga minsan nagka-cancer. Nakukuha niya? Pag sobra, no? You can have uncontrolled growth. Ano naman yung letter B? Sino sasagot nito? Ano yung GLUT4 translocation via TC10? Anybody?
Doc,
Yeah.
yung
Go ahead,
GLUT4
anak.
translocation,
Uh-uh.
nangyayari po siya sa PIK3, AKT
Okay.
independent pathway po.
Okay, so it's not involved in this pathway, no? So, let me just talk about GLUT4. Saan nyo nakikita ang GLUT4? What organs? There are only 2 tissues with GLUT4. Ano yon? John Paul, kanina ka pangitingin ng ngiti. John Paul, ano? Saan may GLUT4? One is
Adipose po.
adipose tissue. Very good. The other is
Skeletal muscle.
skeletal muscles. So these are the only 2 organs that have GLUT4. And GLUT4, as you know, are insulin dependent. Ibig sabihin, pag walang insulin, nagtatago sila sa ilalim. Ngayon, pag may insulin, nata-translocate sila sa plasma membrane. And the translocation of your GLUT4 is the reason why insulin lowers blood glucose levels. Kasi papasok na sila lahat sa cells. Make sense? Okay. Ano naman yung, yun, we talk about GSK3 already, right? So, GSK3 is supposed to, inhibition of GSK3 is supposed to facilitate glycogenesis. Basta at this stage, dapat alam nyo, ano ba talaga yung nag-occur sa well-fed? It's all genesis, no? Except glycolysis. Pag fasting naman at saka starvation, it's all lysis except gluconeogenesis. Maliwanag po? Okay, very good. So, nasa na? We also talk about mTOR2. Bakit mali ang mTOR2? Letter D. Anybody would like to give an answer? Hoy, yung iba dyan, nagtatago sa camera, tatawagin ko rin kayo. Hoy, bukas. Nandyan pa ba kayo? Umuwi na kayo? Sino mag-recite? Ano ang mTOR2? Riley? Martin?
Ma'am,
Ma'am, ano yung mTOR2?
mTOR2 is the enzyme that activates the endogenous glucagon.
Ano?
It's
Are you Riley or you're just Ryle? Martin, go. Okay,
Just
go
Ryle
ahead, anak.
po. For number 8,
Ah,
our
Number
group
7
answered
pa lang.
eh,
Choice D.
choice D.
Choice D. Why is it wrong? You want to go to number 8 na? Di pa tayo tapos sa number 7 eh. May bitin pa eh. O, Christian, tapusin mo muna. Bakit mali yung choice D ng number 7 bago tayo pumunta sa number 8? Christian?
Doc, rinig po ba ako?
Yeah, I can hear you, Eva.
Okay po. Mali po siya kasi hindi po nai-inhibit yung lipid synthesis. Siya po yung nagsisignal ng lipid
Correct.
synthesis sa nucleus po.
Correct. Very good, no? So, whenever you have mTOR2 or mTOR1, these are the sensors of nutrient status. So, nasa-sense niya, kulang na ng ganirega, umawa ang katawan. Ganun lang siya. Okay. Riley, go to number 8 na. Nagmamadali si Riley. Mukhang may date eh. O, go na.
For number 8, activation of SIK2
Okay.
to CBP slash P300 by AKT2 results in the inhibition of its metabolic pathway. We answered B, gluconeogenesis.
Gluconeogenesis. Okay, very good, no? So, for you to remember, SIK2, CPB, or ano nga ba ang CPB? Binding protein, no? This is a transcription factor also that will have to enter the nucleus of the cell in order to stimulate your transcription of your key regulatory enzyme. So, gluconeogenesis, palaging SIK2, CPB, P300. So, those are the associations. Now, in the same manner na mali na yung beta oxidation. Kasi ang beta oxidation, anong transcription factor para matandaan nyo? Letter F. Letter F. FOXO. Okay? Okay. Yung ketogenesis. Pareho lang. No? So, whatever transcription factors will stimulate your beta oxidation, makadugtong sila eh. Correct? Okay. So, lipolysis naman, palaging hormone-sensitive lipase, which is connected to glucagon. Yan, para matandaan nyo. Any questions so far? Okay, let's go to number 9. Group 9.
Hello po, Doc.
Go ahead, Chloe.
So for number 9, the question po is, an effect of insulin results to which correct organ metabolic pair?
Okay.
Our group answered letter A po,
Letter A.
adipocytes
Lahat
increases
kayo nag-answer letter
lipogenesis.
A?
Yes po.
Okay, very good. Sila tinatanong ko, ikaw din sumagot. Oh, go.
Ay, sorry.
Bakit mali ang letter B?
Yung letter B po, mali po siya kasi decrease glucose uptake po. Ang insulin po, it helps facilitate the uptake of glucose into cells, including po yung brain and yung muscle. Kaya
Correct.
mali din po yung letter D.
Okay.
Tapos
So,
yung
dapat
letter
A.
C, po. And then, yung letter C po, the body primarily relies on glucose po. So,
Okay. So, walang glyco.
opo, wala pong glycogenolysis.
Very good, Chloe. Any questions?
Thank you po.
So, when you see questions here, you take note of the organ, no? Palaging take note of the organ which is being asked and the time. Is it well-fed? Is it fasting? Or is it starvation? Okay, number 9. Ay, hindi. Number 10 na. Group 10. Group 10. Sige na. Who wants to answer? From group 10. Hoy. Pakita naman kayo. Paramdam naman kayo. Kung nandiyan pa kayo. Anybody? So this is talking about, as early as now, you have to understand diabetes mellitus is a genetic problem. Okay? The predisposition to develop diabetes is genetic. So, ang ibig sabihin nito, if there is a positive history in your family, then you have a tendency to develop. Lalo kung lamon ka ng lamon, hindi ka nag-exercise, asigurado. Okay? O, number 10. Group 10, anong sagot nyo? So this is talking about an infant, pagkapanganak pa lang, diabetic na siya. Sige na, sagot na. Anybody? Yes, Geraldine.
Good afternoon po, Doc.
Hello.
The question is, why does mutation of the gene coding for KCNJ11 result to neonatal diabetes mellitus? Our group answered letter B, ATP dependent potassium channels are always open. So, if recalled from the first sem na lessons po, when the ATP dependent potassium channels are closed, it causes polarization and therefore opening of the voltage gated calcium channels. Then there will be an increase in intracellular calcium, and then there will be exocytosis of insulin. So nagde-decrease po yung blood glucose. So if the ATP dependent potassium channels are always open, the cascade of events I mentioned won't push through po.
Very good.
Kaya nagkakaroon ng neonatal diabetes mellitus po.
Okay, very good na. So it's not because you want to secrete that the potassium channel should be open. Kailangan close. What controls the opening or the closure of the potassium channel? Which of these two? TX62 or SURI1? Which controls? TX6.2 or SUR1? Oh, sige, para may clue kayo, sulfonylureas are agents used to lower blood glucose. So, basically, it is SUR1, no? That will control the closing or the opening of your potassium channel. Kasi if you look at potassium channel, octamer naman siya. Octamers, no? Madami siyang components. And then you have 4 KIR62 and 4 SUR1. The 4 SUR1 are surrounding the voltage channel. So, para ma-regulate niya mabuti yung opening or closing. So, this is a situation where the baby is born and you get a blood sugar, diabetic na kaagad. So, these patients will be forever on insulin treatment. Kasi palaging naka-open yung dapat naka-close. So, you call this your neonatal. Huh? Any questions? Nasasad na kayo? Pero kung ganun na kayo, oh. Okay ba? Oh, number 11. Group 1. Balik tayo. Number 11. Termination. How do you terminate insulin effect? Kasi halimbawa, wala nang pagkain, hindi pwedeng insulin palagi, right? Group 1, can you talk about this? Anybody? Sige na. Sige na. Sige. Pag sinabi nating signaling pathway of insulin, di ba phosphorylation? Tama? So most of the stimulated hormones are kinases. So in order to terminate, you have to remove the phosphate groups. You call that your dephosphorylation. Medyo magulo, no? But that is the parang summary. Okay. So what did you answer dito? Nalulungkot na si Christian, no? Laki ng problema. Oh, Christian, sagot ka nga, anak. Ano sinagot niya dito? Unmute. Ayan, go ahead,
So,
Christian.
for number 11 po, which of the following factors will result to the termination of the signaling pathway of insulin? So
Okay,
we answered po letter B.
letter B.
The phosphotyrosine phosphatase 1B
Ah,
contributes to the termination of insulin action po. Because
okay.
it also because it would inhibit the activity of RTK po, enabling the insulin to phosphorylate.
Okay, very good. Ano naman si P10? What is the action of P10 para maalala nyo? Why is letter C wrong? What is the action of P10? It is supposed to be an enzyme, diba? Anybody?
So mali po yung sa letter C po, si P10 because dapat po PIP3 po siya to PIP2, not po PIP2 to PIP3.
Baliktad. Okay. Very good. Very good, Christian. Ano? Eh, yung letter D, bakit mali? Bakit mali? Common sense. Exocytosis ba? Dapat?
Endocytosis po.
Endo. Kasi endosomes eh. These are structures that have hydrolytic enzymes. So pag pumasok at hindi nilabas, ma-degrade yung insulin plus the receptor. Okay? Did you get that? Okay. Any questions so far? Ano naman yung letter A? Bakit mali? Anong action ng serine threonine kinases? Bakit mali yan? Anybody? Sabi ko, di ba, dapat ma-dephosphorylate, hindi phosphorylation. So, start from that context. Okay? So, dapat, yan yung hindi siya, dapat dephosphorylation yan. Okay? So, let me repeat. When insulin binds to its receptor, there is autophosphorylation of critical tyrosine units, subunits, and this will now attract other kinases, phosphorylate and activate kinases. So if you want to end the signal, you have to dephosphorylate what was phosphorylated. Is that simple enough for you? Huh? For you to remember by. Okay. So now let's go to 48 hours after full meal. Ano na tayo? Fasting. Fasting na po, no? So what will be the major enzyme here? Insulin pa ba or iba na?
Glucagon pa daw.
Yes, Geraldine.
Glucagon na po.
Glucagon na. Ha? Ngayon, if the organ is the muscle, we have no receptors for glucagon in muscle, so catecholamides na, epinephrine. Okay. So, sagot mo nga ito, anak. Mechanism involved increased synthesis of PepsiK. PKA ito, ha? Kasi sa insulin, PK, B, and C. Tandaan nyo, ha? Okay. Yeah, go ahead.
For
Yeah, go
number
ahead.
12 po, the question was, what is the mechanism involved in the increased synthesis of PepsiK by activated PKA? Our group answered po, letter C, translocation to the nucleus activating CREB, CBP, CRE on DNA.
Okay.
Ito po yung answer namin because activated PKA translocates to the nucleus via the nuclear pores po. And then yung activated po na CREB activates and binds to the CREB binding protein or the CBP.
Okay.
And then the activated CBP po controls the expression of genes with camp-sensitive regulatory elements or CRE on DNA. And then yung magiging result niya po would be the synthesis of new proteins po.
Okay, basically,
Yung
it
four
will,
enzymes was the gluconeogenesis.
yeah, it will be the key enzymes of gluconeogenesis. Talk about the signaling mechanism of glucagon, which is also similar to all the other hormones, no? So, simple lang ito. Simple lang yung cascade mechanism ng glucagon compared to insulin. Okay, ano sagot niyo dito? What did you answer? Group 3. Hello? Anybody wants to answer? Sige, chorus na lang. Ano sinagot niyo?
A po, Doc.
Letter A, Jobirinda. Okay. So, you know that G protein, no, is a trimeric protein. What do I mean by that? It's made up of three elements. Diba? Alpha, beta, gamma. But the alpha subunit has an enzyme activity. Basically, the activation of G alpha stimulatory promotes the phosphorylation. Diba? So, sa halip na G alpha ADP, pag inactivate niyan in the presence of glucagon, maging G alpha ATP, and then it will now dissociate from the beta and the gamma subunits and activate adenylate cyclase. Simple lang. Simple lang yung signaling. So, ano yung G-alpha I? G-alpha I is inhibitory. Kasi yung isa, G-alpha S, stimulatory. This is the signaling pathway of somatostatin. That is the reason why it inhibits insulin and glucagon secretion. Gets nyo? Okay.
Yes,
CAMP will bind and destroy. I-activate mo nga eh. So this statement is wrong, no? Dapat i-activate mo yung regulatory portion of the CAMP, of which you have 2 monomers. So, mali yung GTP kasi dapat activate niya, hindi inactivate. Any questions about number 13?
None po, Doc.
Okay, let's proceed. Number 14. Who wants to answer this? Madali lang ito, no? So, take note of the time, 48 hours. So, always take note of that. So, ang sagot lang dito, either A or B. Tama? But since it is already 48 hours, wala ka ng liver glycogen. No? Because the liver glycogen stores are usually lasting only for 24 hours. So what is the other substitute to produce glucose? It is gluconeogenesis. So when you look at questions like this, look at the organ that is being asked, look for the time kasi that will determine the answer. Okay pa ba tayo mga pare ko, mare ko? Si John.
Yes po, Doc.
Okay, let's proceed. Number 15. So let's now talk about a newly discovered group of hormones we call incretin, of which you have 2 types, right? GLP-1 and GIP. No? And we call them incretins because they are secreted after a meal. So, kailangan oral yung intake. Pwede naman konti lang ang marirelease after an IV. Kasi it is in the small intestines pala, I'm sorry, in the ileum, no? Kaya kailangan kumain. Okay. So what is an action of GLP-1 and GIP-1? Common action. Group 5. Sige na, napapagod na ako. Group 5,
Hello po,
sagot nyo
Doc.
dito. O, go ahead, Anne.
Yung sagot po namin, letter B po, insulin secretion.
Kaya nga siya kabarkada ng insulin, no? Kasi when you eat, no, you stimulate, your incretins stimulate the secretion of insulin from the beta cells. So, hindi pwedeng may apoptosis. In fact, it will decrease beta cell apoptosis. So, palalawigin niya yung buhay ng beta cells. May effect ba siya sa alpha cell apoptosis? Meron ba? Sagutin niyo na ako para makauwi na tayo. Ano? Meron, wala? Paulo, Nicolas. Unmute. Ano sagot mo? Meron bang effect ang incretins sa alpha cells apoptosis? Wala. Oh, sino nagsabi sa iyo? So wala, no? It has nothing to do with alpha cell. So dahil insulin secretion siya, syempre yung glucagon, hindi na pwede. Okay. Now remember, aside from the effect of GLP-1 and GIP-1 on the beta cells, it has also other effects on the other organs of the body. So in the handouts, you will see the effects. Okay, let's go to the last one. Yay! Sino mag-discuss? Kurt. Kurt?
Hi, Doc. Good afternoon po.
Hello, good afternoon. Ang sagot natin dito?
Ang answer po natin is letter D.
Okay.
Dipeptidyl peptidase 4. Basically, ang purpose po niya is to degrade or inactivate incretins po. Yung choice D.
Okay, DBP4, no? So sometimes when you're given incretins, the drug will also include a DPP4. Kasi if you look at the half-lives of insulin, seconds lang yan, no? So you need to delay the degradation. And that is because of, I'm sorry, DPP4. Okay. So are we good? Are we good? Sagot naman kayo. Para naman kayong mga tuod dyan. Wala akong kausap. Good? Okay,
Yes
can we
po.
call it a day?